Hello everyone. This video is an introduction to how to use the NCBI BLAST tool for your genetic research. First, NCBI is the National Center for Biotechnology Information Advances Science and Health by providing access to biomedical and genomic information. The NCBI, or the National Center for Biotechnology Information, provides access to several databases with biochemical and genetic information as well as bioinformatic tools. I will show you how to navigate some of these databases to learn more about specific genes and proteins. We will also take a closer look at how to analyze gene variants and their mutations. Let's start on the main NCBI website. To get there, just type in NCBI in your web browser, and then click on the NCBI website link. The website interface will appear. On the left side of the website you will find a detailed list of resources that you have access to. On the right side, some of the most popular resources are listed, such as BLAST tool, and this what we gonna to talk about in this video. The first stage, we will search for a gene or a protein, so Medline Plus will help us. I will put the link in the description box at the bottom. I will search for Down syndrome. Then here a description of the syndrome and its reference will appear. Here is the cause, which is the important part because it contains the name of the gene or protein that causes this syndrome as it is explained, and it's trisomy 21. Then we will choose the database to search. And write the name of the gene or protein here. Then click Search. Many results will appear. I will choose the first. And show me the summary of the gene. And the genetic context. Here it will show the genomic regions, transcripts, and products. Then we will choose the FASTA format, which is because it is the most common in this format. Nucleotides or amino acids are represented in a sequence using single-letter codes. The nucleotide sequence will appear as shown, a very large sequence. To make it easier, we will click on Run BLAST. Here is the main BLAST page where you can compare the sequence with many other sequences in the database. In the first section, the sequence has been pasted, which is in FASTA format, and it is also possible to download a sequence from a file. In the second section, it is allowed to specify your search settings. I will choose to search in a standard database. To narrow the search scope, we will search for a sequence in a specific organism, which is the human. Then we will click on BLAST. It will take time to loading. On the top left of your BLAST report, you will find a summary section with information on different aspects of your search. On the top right, there is a box that allows you to filter your results based on organisms, percent identity, or e-value. Below the top section, the BLAST results are shown. There are 4 different tabs called Description, Graphic Summary, Alignments, and Taxonomy. Each tab presents your search results in a different way. The Description tab, which is the default tab, contains a summary table of hits found by BLAST. Each row in the table represents one database sequence that BLAST identified to be similar to your query sequence. You can use the checkbox in the top left corner to select all or none of the sequences. The table provides following information: the description or title of the matched database sequence, The maximum and total alignment score for the database sequence. The query coverage, which is the percentage of the query sequence that is covered by the database sequence alignment. And the expect, or e-value of the database sequence. The e-value is the number of expected hits of similar score that could be found just by chance. The lower the e-value, the more confident you can be that the result sequence is not a random hit. You will also find the maximum identity, or the highest percent identity of the database sequence. And the accession number of the database sequence. The accession number is a unique identifier given to a specific nucleotide or protein sequence in a database. The Graphical Summary tab provides a graphical overview of the alignments between your query sequence and the database sequences in your BLAST results table. The query sequence is shown at the top. The colored boxes underneath correspond to regions in the query that have sequence similarity to the database sequence. The boxes are color-coded based on their alignment score. Red bars means most similar sequence. Pink bars matchless good. Green bars not an impressive match. Blue bars worst score. In this sequence, there is no blue bar. Black bars bad hits. In the Alignment tab, you can view detailed information about the alignments between the BLAST hits in the Description tab and your query sequence. The alignment shows how well the sequence matches the subject sequence. The top row of letters corresponds to your BLAST query sequence. The bottom row of letters corresponds to the matched subject sequence in the database. Vertical lines connect nucleotides or amino acids that are identical in both sequences. The sequence identity above the alignment tells you the number of identical bases or amino acids between the query and the subject sequence. And in the Taxonomy tab, the report shows us either by the lineage or the organism, and we chose the human being in the beginning or the taxonomy. That's all for this video. I hope you all like it. Don't forget to subscribe, like and turn on the notification bell.